Okay, welcome to our webinar organized by the PurPest project on VOC sensors for early pest and pathogen detection insights from the PurPest project. My name is Justus Wessler. I am the chair for agricultural economics and rural policy at Wageningen University, and I will be the moderator for this webinar this afternoon. Now, before we start, some housekeeping rules. So please all mute your microphones. If there are questions and comments, please use the Q&A button in Teams for raising comments and questions. Now, how we will organize the webinar is we will first have an introduction to the project by Andrea Ficke. She is Senior Researcher, Division of Biotechnology and Plant Health at the Norwegian Institute of Bioeconomy, and at the same point in time, the project coordinator of PurPest. And then we will have 5 presentations on the important pests that are addressed within the project. This includes the fall armyworm. There we will have a presentation by Amandine André. She's postdoctoral researcher at the Fundamental and Applied Research in Chemical Ecology Lab at the University of Neuchâtel in Switzerland. Then on Phytophthora ramorum by Thomas Jung, head of the Phytophthora Research Center at Mendel University in Brno in Czechia. Then on the pinewood nematode by Jorge Faria. He's researcher at the National Institute for Agrarian and Veterinary Research in Portugal. Then on the cotton bollworm by Jörg Romeis. He is head of safety research group at Agroscope in Switzerland. And then on the brown marmorated stinkbug by Jana Kollatz, research associated at the biosafety research group, also at Agroscope. After these presentations, we will move into the technical issues, and there we will have a presentation on how VOC sensor devices work. And that will be done by Damien Bazin. He's from the analytical department of Arimotech at the Chromototech group in France. And then finally, we will have Stelios Katakis. He will talk about the economic implications and the economic benefits that early damage control pest identification has to offer us. Now, with these introductory words, I would like to hand over to Andrea Ficke for introducing the PurPest project. Andrea, the floor is yours.
Thank you very much. I think I will just start sharing my screen and actually go into the presentation mode. Thank you very much for giving me the opportunity to introduce you to the PurPest project. The PurPest project is a 4-year project basically funded by the European Union under the Farm to Fork section, and it really addresses our objective to go towards pest sensors for pest and pathogen detection. Why would we really care about that? Well, as we transport and import and trade a lot of plants and beautiful flowers, we are also allowing a lot of pests and pathogen into our countries. It's a highly volatile area where you have a very difficult area where you can not really see the pest and pathogen on the different plants that you're importing. Here is just an example. When you're opening a container that is transporting some plants, it's full with different plants. Here you have 2 here, but you could have 500, you have a lot of different varieties of plants that can be imported. They always travel with a plant passport or sometimes even a plant health certificate. But this paperwork, even though it is checked, it is usually not reliable. That means it does not reflect the actual plant health status of the plant. We know that the plant health inspectors in the different countries are using some random sampling to see if those health requirements are fulfilled, and often it is not. But in any case, they cannot really check all the incoming plant shipments. So they might visually inspect only 3% of the incoming plants that are traded. So 97% is not really inspected, and we do not know what comes in there. The reason for that is that it's just the sheer volume. It's impossible to inspect each and every plant and each and every shipment. We don't always have the competence to really recognize those pests and pathogens that are coming into the country. It is sometimes difficult to motivate people, especially if the plant health inspectors are employed by those people that are importing the plants, because why would they really want to find something that will just cause them trouble and stop the shipment? And the detection success is really low if you just look visually for some eggs, some phytophthora that are in the soil, nematodes that are in the plants. You cannot really detect them visually. So that is very low success. So with our project, we want to facilitate the detection of pests and pathogen, and not just in like one container at a time, but actually at several containers at the same time. And the way that we want to approach this is not by visual inspection, but by chemical inspection. So each of the plants that are coming in, each plant that is growing, each living organism actually emits smells, volatile organic compounds. And these smells can be characteristics because they change with the stress status. If a plant is attacked by a certain pest, it will change its chemical odors, the odor that they're emitting. And in that regard, we think we can actually detect the different pests that are on the plants that are coming in by detecting these specific volatile organic compounds. The pests that we are focusing on to sort of show if this concept is working are very diverse. We're working on the Phytophthora ramorum and Franz group. So I call that it's a whole complex, but there are many different Phytophthora that are attacking plants coming into the countries and are traded with the plant material. We have the fall armyworm. We also have the pinewood nematode looking at, and we have the brown marmorated stink bug, plus the cotton bollworm. So we have 5 very diverse pests that we want to use as model organisms to test if this approach is going to work. We have come pretty far in our project. We're now in the 4th year, basically, and we have come far enough that we have a concept. We have a working sensor system prototype that Damien will talk about. And now it's the time when we really need the input from stakeholders. We want to know basically, will it be useful? Can it be, how can you use it? What is most important to users, not just for the plant health inspectors, but also for the people working in nurseries, they're receiving the plants, or those that are sending off the plants. Who will actually use this? Are there other people other stakeholders that could think that this is useful? Storage control could be important there as well. And we would really like to know what concerns you might have. What are the barriers? What is the most important thing for you if you have a detection platform? Is it the robustness? Is it the pricing? Is it how sensitive it is? So these questions I hope you can address and you can discuss a little bit later. And with that, I think I will open the floor for the pest experts. And I think we start with Amandine talking about the fall armyworm so that you get an idea about the model pests that we're working with. Thank you.
Thank you, Andrea, for this concise overview about the PurPest project. And now we look into the first important pest to be covered, the fall armyworm. Amandine André, please, the floor is yours.
Thank you very much. Thank you, Andrea. Thank you, Justus. And good afternoon, everyone. It's my pleasure to introduce this session with the first pest of the project, the fall armyworm, and to explain you a little bit more how we intend to exploit pond volatile for the early detection of this major pest. So the fall armyworm is a highly invasive pest, a polyphagous specialized on mice, and is causing massive economic losses. So the adult females are laying eggs on the leaves of the plant, and after hatching, the young caterpillars begin right away to feed on the plants, which is causing a lot of damages, as we will see in the slides that are following. So the fall armyworm is originating from the American continent, and due to its exceptional capacity to fly over hundreds of kilometers in one day, it had invaded Africa, Asia, Australia, and have been detected for the first time in Europe in 2024. With the climate warming, there are some predictions that the fall armyworm might establish along the Mediterranean basin. Here are some pictures of a maize field in Africa that have been invested by the fall armyworm, and we can really see the high damages that it causes. And in Africa, it's been estimated around a $10 million annual loss for the whole continent just for the maize production. To fight the fall armyworm, the farmers are spraying the fields with a lot of insecticides, which is problematic not only for the consumer, but also for the health of the farmers, because as we can see here on this picture, the farmers, especially in Africa, are wearing very little to no protection at all against those pesticides. So we have to find a solution so we can reduce the amount of pesticide employed against this pest and also find some biocontrol ways. But to do so, we first have to understand how we can detect this pest as early as possible. And to do that, we were inspired by nature and especially by this parasitoid wasps, which is laying her eggs inside the caterpillar. And in the early 90s, it was thought that these wasps were attracted by the volatile organic compounds emitted by the caterpillar itself. And during his PhD, the Professor Turlings found out that actually the wasps was not attracted by the caterpillar first, but by the damaged plants. So the wasp was attracted by the volatile organic compounds emitted by the plants, and that she used to find her host and then lay her eggs inside the caterpillar. So we can see that as the way that the plant is calling for help and recruiting those natural enemies to get rid of her pathogen. So we started to collect the volatile organic compounds from different plants, so undamaged plants, as we can see here. So we record what we call chromatograms, which is an overview of the chemical composition of the volatile compounds emitted by the plant. And we can see in the undamaged leaves that there is very little peaks on this chromatogram, just our internal standards that we add. After a couple of hours, or just right after the larvae starts to feed on the plant, we can see that we have already some peaks that are emitted by the plant. And after a couple of hours of infestation, the chromatogram looks totally different, and the plant is emitting an odor that we intend then to use as a fingerprint in order to detect if the plant is damaged or not. And the idea also of the PurPest project would be to develop these sensors, and they could be mounted on other sensor module on rover that could run over the fields and detect if the field is attacked by pests or pathogen. In our lab, we use another type of sensor that's called proton transfer reaction time of flight mass spectrometry. I'm not going to enter too much into details. We can see on the left side of the picture that it is still quite a bulky device, but it's been miniaturized in order to fit on the rover and to go onto the field. But thanks to its high mass accuracy and high sensitivity, we are able to detect in real time the odor profile of a plant that is damaged by a caterpillar. We can see here in red this chemical fingerprint, and in black the chemical fingerprint of the undamaged plant. And everything can be detected within less than 10 seconds. We use this device and we wanted to know if not only we can detect if the plant is attacked, but if we could differentiate if the plant is attacked by different kind of pests and pathogens. So we did an experiment with the beet armyworm, another type of Spodoptera, the fall armyworm, and also fungal disease, the corn leaf blight, and of course a control. And then when we modelized the chemical fingerprints of the different plants attacked by the different pathogens and pests, we can really see that the chemical fingerprint is unique, and we have a good differentiation of the different pests or pathogens that are attacking our plants. We took also this device outside. Here it's just in front of the building, where we measured single plants in an outdoor environment, which is much more challenging because of all the other that are around. And we tested if the device was still able to differentiate between a caterpillar damaged plant and a control plant. And using machine learning, we found out that we had a very good accuracy of prediction. So the machine learning was able to tell us, okay, the control plant is a control plant or the damaged plant is a damaged plant. And the accuracy was very high at 96%. We also took this into a mice field. There, the accuracy tests decreased to 70%, but we are convinced that with further research and development, we can achieve higher degree of accuracy of prediction. So to conclude, other sensors can be used in the field for the early detection of major pest insects in real time. And I thank you for your attention, and I hand over to the next speaker. Thank you.
Many thanks, Amandine, and very nice introduction to the fall armyworm and the cry of plants for help, which also illustrated some of the interesting features of biology. Now I'd like to hand over to Thomas Young, and he will introduce Phytophthora ramorum. Please, Thomas, the floor is yours.
Thank you, Justus. I will introduce to you Phytophthora ramorum, its origin, the diseases of forests and ornamental plants, and the importance of the nursery pathway in spreading this important pathogen. This is a simplified phylogenetic tree, which shows you that Phytophthora belongs to the oomycetes in the kingdom Chromista and is not at all related to fungi, which are here. Phytophthora are related to brown algae and diatomea, not to fungi. These are the structures of Phytophthora ramorum. You can see in the upper row sporangia, which is the major infective propagule, and the sporangia of Phytophthora ramorum, they break off from the hyphae, from the sporangiophores, and fly through the air like fungal spores, and they also release zoospores that are infecting the leaves. Phytophthora ramorum also produces vegetative resting structures, chlamydospores, which you can see here, and if both compatibility types of the pathogen co-occur, then they also form the sexual structures and the oospores, which can survive over many years. The origin of Phytophthora ramorum lays in East Asia. We found it in northern Vietnam around the Fansi Pan Mountain and on the Japanese islands Kyushu and Shikoku. In total, 12 lineages of the pathogen are known, and only the EU1 lineage is currently present in the EU. All the other 11 lineages are on the A2 quarantine list of the EU and EPO and are regulated. This is a typical forest where Phytophthora ramorum comes from. It's a subtropical cloud forest at the Fansi Pan Mountain in Vietnam. This is an example of a mountain forest in Shikoku Island, where Phytophthora ramorum and its closest relatives, Phytophthora lateralis, also a quarantine pathogen in Europe, and Phytophthora foliorum co-occur. And this is on Kyushu Island, a forest where Phytophthora ramorum and the other quarantine pathogen, Phytophthora lateralis, co-occur. The 12 lineages of Phytophthora ramorum are separated from each other since millions of years, and they have developed lots of differences. You can see here in this panel that the colony morphology of the 12 lineages is very different from each other. They are also behaviorally very different from each other. You can see here that the radial growth at 28 degrees Celsius in culture is very different between the different lineages, and 3 lineages here on the bottom don't grow at all at 28 degrees. Other lineages like the North American NA1 lineage grow very slowly. Also the European EU1 lineage is growing slowly, and other lineages grow very fast at 28 degrees Celsius. Phytophthora ramorum is the causal agent of sudden oak death in California. The pathogen infects there the understory shrub bay laurel, and sporulates on the leaves. From the leaves, the sporangia then spread onto the stems of oaks and 10 oaks and cause there these bleeding canker lesions. And since they cause up to 2,000 bleeding lesions on the bark of an individual tree, the trees succumb very quickly and dry and wilt and die within a few months. In Europe, since 2010, the pathogen is causing a similar disease on large species, European larch, Japanese larch, and the hybrid larch between these two large species. Here also it causes on stems and on branches bleeding cankers, resinous cankers, which you can see here red underneath the bark, and the trees die very quickly. The United Kingdom is hammered by this disease, and they had to remove millions of trees in eradication efforts, which you can see here, clear felling of the infected stands. In whole of Europe, also here in Central Europe and in North America, it is also a major pathogen of rhododendron species causing leaf and twig blight. And you can see here these typical necrotic lesions on the leaves and here on the twigs of rhododendrons. And rhododendrons from the nurseries are also the major means of spread of the pathogen across the continents. We did a nursery survey in 2016, which we published in 2016, but it was actually a survey running over 20 years. And you can see of these 1,993 isolates that we got from the nurseries, 10% of the isolates in the nurseries were Phytophthora ramorum. That means that the European nursery trade is heavily infested and contaminated by this pathogen. We also tested 2,525 planting sites across Europe. And also here of these 2016 isolates, 3% were Phytophthora ramorum. So the pathogen is very widespread. Thank you for your attention. This was a very brief talk about Phytophthora ramorum. The situation with the volatiles is quite complicated with Phytophthora, but we have also very interesting results from there, which we hopefully will publish soon. Thank you.
Yeah, Thomas, many thanks for a concise overview about the challenges caused by Phytophthora ramorum. And we have seen some of the interesting plants like the leech and the chrysanthemum are affected. And that is something that might be of concern for many of us, and in particular when we talk about the rhododendrons. Now I'd like to hand over to Jorge Faria. He will talk about the pinewood nematode. Jorge, the floor is yours.
Thank you. Good afternoon, everyone. My name is Jorge Faria, and I'm going to talk a little bit about the pinewood nematode, which is a pest studied by the PurPest project. So the pinewood nematode is a phytoparasitic pathogen that can be both mycophagous and phytophagous, which is rather unique to the genus. It generally inhabits dead or decaying wood, feeding on fungi, but the problem arises when it finds its way into the inner tissues of pines. It can parasitize pine and kill pine very quickly. It's believed to be endemic to North America, where it has no huge impact in forestry, so there's not a big expression of the pine wilt disease. And in the beginning of the 20th century, it was found in Japan, where it's since then been causing extensive damage to pine forests, and it quickly spread to other Asian countries. And in 1999 was discovered, first detected in Europe, in Portugal, close to a shipping port, and it's also spread to Madeira Island, still in Portugal, and to border forests in Spain. And unfortunately, late last year, it was first detected in France, which means that it's slowly spreading throughout Europe, alarmingly. So pinewood nematode causes pine wilt disease that has a rather sophisticated infection cycle involving many organisms. Generally, the pinewood nematode, as I said, lives on dead and decaying wood, feeding on fungi. In what we call the propagative phase, it quickly goes through all its stages of its life cycle, and we can get, generally it's the stage we use in the lab, because we can get a lot of population really quickly. The problem is that these sites are also used by the female Monocamus genus for oviposition, to lay its eggs, and then the eggs go through all its stages, the larvae, pupae, and young adult. And when it reaches this young adult stage, it becomes very attractive to the pinewood nematode that goes into what we call the dispersive phase with huge morphological and biochemical changes, and it colonizes the insect through its respiratory system. And the insect, when it emerges, it's really hungry, so it tries to find the young tree branches and starts feeding on them. And when this wound is opened, the nematode senses the presence of a susceptible pine, and it goes from the insect into the pine and starts feeding on its internal tissues, on resin canals, and on vasculatory system. And this causes, of course, interruptions in the water column. And if the water doesn't reach the canopy, the first symptoms of pinewilt disease, the first visible symptoms of pinewilt disease can be seen. That is needle yellowing and wilting, and this is more or less the trees that we get in the field. And when the trees are in this stage, they become susceptible. There is no recovery for these trees, and they become susceptible to opportunistic fungi and bacteria. And the nematode switches back to its mycophagous feeding habit, and the cycle goes on again. So you can see it's rather sophisticated with many organisms, and what we don't know is how these different organisms communicate. We believe it's through the emission of volatiles, so we still don't know if the nematode itself produces volatiles, which chemicals or volatiles is the insect producing in this specific stage to trigger the change into the dispersive phase of the nematode. What volatiles are emitted by the pine that attract the nematode, so that induce infection. And even if there's volatiles being emitted by infected material, wood material, that we can use to detect the nematode. This is the aim, one of the aims of PurPest, to try and figure out if these volatiles can be used for early detection. So the impacts, pest management strategies that can be used, that have been used, are the creation of these buffer zones where we eliminate trees to try and control insect flight. This was seen to be ineffective. Also, the installation of insect traps and the use of insecticides to try and control insect population. And heavy investment has been made on monitoring the pinewood nematode, and any infected plant is quickly cut down and destroyed to try and prevent dispersion of the nematode. Any wood was used for export has to be treated, and there is heavy regulation on wood trade, and this, of course, has affected the wood dependent companies. And we can use trunk injection of nematicides, although this has to be in a limited number of pines, of course. So this is what's been the greatest impacts are the money you have to invest on these management strategies, and of course on the wood trade in our country. So thank you all for your attention.
Thank you, Jorge, for this very nice introduction to the challenges we are facing from the pinewood nematode and what this may mean for the trade and exchange of timber products in the European Union, highlighting the importance of having better control methods. So thanks a lot. We now move to the next pest that is of relevance, and that is the cotton bollworm. And I hand over to Jörg Romeis.
Thank you, Justus. Welcome from Switzerland. It's my pleasure to provide you a short introduction into the cotton bollworm, Helicoverpa amagera. For the non-entomologists among us, just a brief look at the life cycle of this species. So you have the adult moth on top, It flies around, it mates, it lays eggs on the plants. From these eggs, little caterpillars emerge, and then they develop through 5 or 6 instars, depending on the climate and the geography. This is the feeding stage, which causes the damage, and then eventually the large larvae, they go down into the soil, burrow there, develop into pupae, and then from there the adult mosses emerge after a couple of weeks. The species is known as an old world species, so it occurs or originates from Europe, Africa, and Asia, and Australasia, as you can see on the map, but it has spread to northern climatic zones in Europe, but also to Central and South America in 2013. In Europe, it has been reported basically in all member states, but it only persists throughout the year in the southernmost parts, in the Mediterranean areas where the winters are not so cold. We know that this species has a very strong flight ability, so it can travel northwards during summers for up to 1,000 kilometers. And so therefore, in many parts of Europe where the species cannot overwinter, we still find transient populations causing damage during the summer months. Interesting of relevance for our project, PurPest project, is that in some parts of Europe, in particular Norway, United Kingdom, this species is still seen as a quarantine pest because it hasn't yet established there, and therefore we also need to make sure that it doesn't reach these areas and can be detected early on. So what makes this species such an important pest? First of all, we know that the adult moths are attracted to flowering plants because they feed on the nectar, on the flowers that the flowers provide. What makes it such a damaging pest is that the larval stage feed on the, or preferably feed on the reproductive structures because they are particularly rich in nitrogen. So it's the reproductive structure, but also the growing tips. The species is extremely fecund, so they lay hundreds of eggs. A single female can lay hundreds of eggs. We know that it can live on a lot of host plants. So the latest number I found is 180 host species from 45 plant families. So it can live on a lot of things. It can fly large distances, as I have mentioned before. It has a facultative diapause. So if the climatic conditions are not ideal, it might just stay in the pupal stage for quite some time and just emerge when the weather is right again. It has a very short generation time. So in tropical regions, we can have 11 generations in a year. Imagine 11 generations means a lot of larvae causing damage to a lot of crops, and it is increasingly difficult to manage because the species has developed resistance against almost all major insecticide classes. Here are a few pictures. So what you can see is the larvae feeding on the reproductive structures, cotton on the right hand and chickpea on the top left. So these are the parts that we would like to harvest. So of course the damage is large. And as you can see in the left lower picture, the larvae also occasionally feed on leaves here on cotton, but this is by far not as damaging. And I have also added a picture on the right hand. So the species is, interestingly, it's quite difficult to culture despite its high fecundity and reproductive ability. They are very aggressive. The larvae can feed through plastic, so keeping them in the lab requires some effort. So you need special strong cases to keep them. So what we have done within the PurPest project is to look into the economic impacts, or what we know about the economic impacts and about the ecological impacts of this pest. And just a few words here. If we look along the invasion process, there is some economic impacts of the spread of this insect on the various steps. So at the invasion or transport stage, of course, there is costs involved in investing in phytosanitary measures. You need to invest in surveillance, trapping, detection, and identification, to prevent the species from establishing in an invasion area. You need to, it comes at costs to potentially eradicate the species once it has been detected, and once the species has established and causes its damage, of course, that comes at a cost. What we also, one conclusion of our research, and this is what fits with the purpose of the PurPest project, of course, is that if we can, it is economically beneficial if we detect and eradicate the species as early in this process, of course, as possible. We have also looked into the literature to look for ecological impacts of the species. I think what is most important maybe here to mention is that the species does not only cause damage to the crops, but also to native non-crop plants, wild plants in the invaded areas. And of course that it interferes with the established food webs with other species occupying the same ecological niche like Helicoverpa, for example, in the New World. This is published, okay, and you have the reference below. Just a few words at the end on the management of the pests. There is established methods using pheromone and light traps to detect and monitor the spread of the species and the presence of the species. These traps are also used for mass trapping with medium success, I would say. A well-established control method is transgenic plants, BT, maize, and cotton. Australia and China are to be mentioned there as an example. Chemical insecticides is still the primary management tool despite the problems with resistance. There is some biological products, plant extracts and virus preparations that have some effect, and biological control in particular with egg parasitoids that kill the egg stage is commercially available in some parts of the world. Thank you.
Jörg, thanks a lot for illustrating the challenges that we are facing with controlling the cotton bollworm. And nice to know that it is not only or maybe not so nice, but it's going beyond cotton, right? So it's also something that we as Europeans should be concerned about when we think about ornamental plants and biodiversity. Now I'd like to hand over to the next presentation by Jana Kollatz. She will talk about the marmorated stink bug.
All right, thank you for the introduction. So the last species we are talking about is the brown marmorated stink bug, Haliomorpha halis, or BMSB. So BMSB is a highly fecund plant-feeding pentatomid. It develops from eggs through a number of nymphal instars into adults. Here in Switzerland, BMSB usually produces only one generation per year, but in warmer climates, up to 6 generations per year can develop. The species is native to Asia, and from there, multiple introductions have happened into North America and into Europe, but also introductions from North America then into Europe have happened. So one pathway of introduction are the aggregations of overwintering adults on various materials such as crates and dunnage. And then an important pathway are immatures on fruit, vegetables, and nursery stock. Adults can also travel quite some distances hiding on cars and trucks, and there are also active flyers covering distances of a couple of kilometers within a few days. BMSB is quite firmly established in Central Europe, so in particular Switzerland, Germany, and Italy. But it is still spreading within other invaded countries, so a lot of the countries that you can see on the right here. And there are even more areas worldwide predicted to be suitable for establishment, and BMSB is also regularly intercepted at phytosanitary border controls. Quite interestingly, the damages of BMSB are usually only reported a few years after the introduction of the species into a country, but then they become quickly quite significant. And this is because BMSB feeds on over 100 host plants. Among them are pear, peach, apple, pepper, tomato, soybean, hazelnut, which are all quite strongly affected. And in particular, damaging is early season feeding, which happens during a critical growth stage of the plant fruit. So on the right, you can see strong deformations in peaches and on pears, where this early feeding has happened. But then there's also later season feeding, which leads to spots and to corky tissues in the fruit, which reduces their quality. So, as Jörg mentioned, economic impact can happen at different stages of the invasion process. So there are trade restrictions and phytosanitary measures that have to be taken before export, for example, to Australia and to New Zealand during particular seasons. There's also costs around early detection monitoring, even though there are no specific cost reports for BMSB. But then at the later stage, where the species is established and causing impact in the fruit crops, we have quite some specific reports for BMSB, so about 356 million losses in pear, peach, and nectarine crops in Italy were reported in 2019. There are also costs of $37 million in apple in the mid-Atlantic US. Insecticide costs are reported to come up to $3,275 per hectare per year. And in addition, we know BMSB as a household nuisance due to their aggregation behavior in fall. And then there's also ecological impacts reported. So BMSB does not only feed on crops, but also on native non-crop plants. It competes with native pentatomid species. There are impacts from management, whether this is conventional management or biological control. And there are effects on native parasitoid networks. For example, when native parasitoids lay their eggs into the eggs of brown marmorated stinkbug, but the parasitoids then cannot develop. There are a couple of different management measures against brown marmorated stinkbugs. So there are pheromone traps used, blacklight traps, and visual inspections for detection. 16 active substances are registered against BMSB in Europe, such as deltametrine, lambda-cyhalothrin, acetamiprid, and spinosad, which all have impacts on the environment. There's biological control using native and non-native parasitoids, and also traps and protective nets are used. So thank you for your attention.
Yes, thank you, Jana, for the introduction to the brown marmorated stinkbug. And again, we can see that that pest affects a wide range of host plants that are of importance within the European Union. Now I'd like to hand over to Damien Bazin. He will talk about the technologies that can be used for identifying the pest. So to the audience, please use the Q&A button in Teams if you have questions or comments. But now I'd like to hand over to Damien. Please, the floor is yours.
Thank you very much. So in this presentation, I will present you the concept, use, and the development state of the instrument which has been developed within the PurPest project. As it has been introduced by Andrea, the goal of this project is to identify some of the volatiles which can be emitted by the plants so that we can clearly identify the pest-infested plants. And the goal of this system is to be used either directly in the field or at import control. In this project, the biologists have used this big machine to actually characterize the VOCs which are emitted by the plants. So here we need to make a system which can be as powerful as the the name of the system is a TDGC MS system, but which has to be very small, which can be deployed in the field. And ideally, it has to be so light, small, can run on battery, and also can be used with either a tablet or a mobile phone. Why it is a very big challenge to analyze these molecules, these very specific molecules which are emitted by the infested plants, is because the volatile organic compounds are actually emitted by biogenic sources. A lot of VOCs are naturally emitted by biogenic sources, even though they are not infested by pests, of course. And the VOCs can also be emitted by anthropogenic sources. Plus, when these molecules are in the atmosphere, they might undergo photochemical oxidation and modification. So actually, at the end, when you measure ambient air, you have hundreds of molecules of VOCs which can be present in the atmosphere. And the goal of this project is to be able to identify these single molecules which are tracers of infestation of plants. So for that, we have developed a system which is called the SSP, the Sensor System Prototype. The first step of the development of this machine was to develop electronic boards which could actually support all the newly developed sensor of this project that could eventually detect precisely and quantifies precisely the specific VOCs emitted by the plant. This has been done in the project, and you can see this small board which has been designed, which can actually be used with one or multiple sensor system. It can run on battery, and actually this is the electronic plus the computer, so it's a very tiny electronics. After that, we had to develop what we call the firmware, so the software which is embedded into the electronic, and also the software. We need a software which can be very easy to use, because again, it needs to be used by non-specialists, which can take the machine and just press the button and then start the analysis to have an idea if the plant is infested or not. To show you the way the system works, I will show you a very short video. So you can see the interface. Here it's used on a tablet, but it can be also used on any mobile phone. You have only a few buttons to click, and here, sorry for the sound, I mute. Here you see that you just need to select the method that you need to start, and you go for the analysis, and then the analysis starts. And if you need to visualize the results, you just go to the last results, and it will directly display the last result, which has been saved on the machine. So the software has been thought not to be complicated. We wanted to have something, again, so that any end user, even non-specialist, can use it. And here, this is a typical chromatogram of ambient air when you see a 41 ppt peak of benzene, so a few nanograms of benzene in ambient air. This is what the machine actually can detect. So the firmware and software has been developed, quite friendly, and after that we need to work on the hardware integration. So again, the system needs to be very small. It can be just held by a single person. So first in the project, we worked with this 3D representation of the machines. So where we actually work on the integration on every single component. So here I will not enter into detail, but we have here the detector, which has been selected as the best one among the different newly developed detector. Here we have a pre-concentration unit, which actually help to gather the VOCs before they are injected into the machine, and here the analytical column. So we first make the design of the machine, see if the way is correct, so if it can be handled by a single person, and then at the end it looks like this with the system which is here and the small tablet to carry the system. So just to give you an idea of the size of the machine, again, sorry, I like the videos. So here you see just a single user, which is actually putting in his bag the tablet, and he will just take the system with him so that you have an idea how big it is. Okay, and he will just go outside to just show you how it can be done in the field. So of course, we work a lot on the system so that it can be used in different conditions. So in cold weather, in hot weather. So this is all the work which is which has been done and it's still currently done. So we try to improve this, the capacity of the system actually to have the same level of performance, whatever the weather and external condition. So here you see that the user has to drop the machine on the floor. He will take the tablet to have a look at the system. And he will just take here a small Teflon tubing to actually install it and put it to the plant. So that's how the system actually is now. This is how it is evaluated. It's going to be evaluated actually from next week again in Czech Republic. So this is the very same system that you can see here on the video. So that was the hardware integration. And then for the analytical development, I will show you some of the results that we obtained already during the intercomparison. So just to show you that we have actually within this project evaluated different type of analyzer. That was the first prototype which has been designed, which actually didn't give the expected results. We didn't have enough sensitivity for the analysis of the desired molecule. So we had to switch to a bigger unit. And actually, most of the results which I will show you are made from this bigger unit. And then when we realized that all the analytical components of these units could be actually powerful enough to find the molecule of interest, then we miniaturized what was inside this system to a smaller system, a portable system. So we performed already 4 intercomparison campaigns, and I will focus on one mainly, the one that has been taken, the first one that we did in Switzerland when we actually characterized the infestation of mice plants. 2 setups were characterized. Here, the glass bottle, where the mice plant is inside the glass bottle, we flush with the Euro air and we see if we can analyze the presence of the pests. And here we try to simulate, let's say, closer to field condition where we put into such type of boxes different plants and we see if we can characterize the presence of the specific VOCs. Here is a typical chromatogram. This is the type of result that you obtain with this machine, where you have in the x-axis here the time and here in the y-axis the intensity. And when you have a peak here, it means that you identify a single molecule eluting from the system. What we can see if we zoom into this part here is that actually when we have a non-infested plant, we have a single peak here, and then when we have an infested non-infested here, we have a single peak. And we have infested plants here, we have a very big peak that appears here, which is a molecule called DMNT, which gives a very clear signal that we are identifying a molecule characteristic of the infestation of the plants. What was interesting to see is that here it's a graph where you have 3 different things analyzed on this graph. So here you have the time and the on the x-axis, sorry. And here you have the DMNT area. What you see is that in the white zone here, we were measuring ambient air. And in ambient air, you see that we have absolute 0, which is measured. And then in red here is what we put the system in the presence of an infested plant. We see that actually the system detects DMNT. And then when we put a non-infested plant here in green, we see that we measure nothing. So actually we can make the difference between ambient air without no plant, ambient air plus a non-infested plant, and then ambient air with an infested plant. And we can clearly see the difference between infested and non-infested using our system. Here is just a representation so that we can get also the time resolution of the emission of the VOCs emitted by the plants. So here we have 3 molecules, nonanal, DMNT, and benzyl acetate. The first one, nonanal, is actually always emitted. Wherever the plant is infested or not, this molecule is always present. But then DMNT and benzyl acetate are produced then only in the presence of the pest. One interesting parameter which has been observed in this project is that actually at night, the plant stopped emitting completely, which means that we cannot characterize if the plant is infested or not when it's dark. We need light to make sure with this plant and this pest at least to make sure that we can identify the presence of the pests. Also, to give you an idea of how difficult this is to measure these molecules, here when we measure in the cages, the peak of DMNT is quite small. So this is approximately 0.07 microgram per cubic meter. Just for you, in the total VOC in ambient air range from 10 to 1,000 microgram per cubic meter, which means that we need to make a very big differentiation between all these VOCs to make sure that we'll actually spot these very specific VOCs of interest. So that's why it highlights how difficult this is to make this tool. Also within the project, here I just show some chromatogram, but we have developed some very nice program which allows automatic identification just by looking at the profile. So we use some AI software which can directly get the result from the machine, and we don't need any expert or whatever on the system. The system will learn itself to differentiate the healthy and infested plants. And thanks to this machine, we actually checked that with manual let's say identification performed with classical analysis, and with this machine learning, we obtain actually very similar results, knowing that, of course, the machine learning allows for much faster analysis than the manual integration. So we did other evaluation for other pests. So for some of them, as I showed for the maize plant, we cannot say that we get almost a total success with the evaluation which has been made. And for other pests, we got, let's say, some partial success. So for example, for peaches, it worked quite well with BMSB, while we got not so good results with apple, pear, and soybean. But for this, we realized after the evaluation that perhaps we didn't really focus on the right VOCs, that maybe there are some more promising ones. So perhaps we will do some other evaluation to even increase the capability of the system to identify all the desired pests. And we will make some other evaluation and Mendelew, INIAV, and NIBIO to have all the pests evaluated and plants. So at the end, so what we have done within PurPest project is that we have developed the SSP, the sensor system prototype. We have performed the mechanical design and the manufacturing. We have developed the electronic and software, and we have already performed 4 evaluation campaigns where we show that the capability of this new tool to be used for the identification of infested plants, and we have planned already for additional evaluation campaigns to evaluate the system on all of the plant and the pests. Thank you very much.
Damien, many thanks for a very interesting presentation about the technologies that are available for detecting pests and diseases using VOCs. There are questions that have been raised in the Q&A. May I please ask the presenters to have a look at the questions and, if possible, also already starting to write a response to the questions that have been raised. We may later come back to those questions during the discussions part of the webinar. But now I'd like to hand over to Stelios Katakis to talk about the socioeconomic implication. Stelios, the floor is yours.
Yes, good afternoon. Thank you, Justus, and thanks to all the preceding speakers. Very nice presentations. So today my talk is divided into two parts. The first one is the assessment of the potential direct damage costs of 2 examples of the PurPest target species. And the second one is the adoption potential of the sensor, the VOC sensor, among European nurseries. For the first part of the talk, we basically accounted for the potential establishment of the species, meaning that we consider also the climatic suitability and the host availability to reach and run the economic models. And we also use the radial range expansion model in most cases under different scenarios to get a bandwidth of different how the spread basically influences the potential direct damage costs. I will start by the fall armyworm. As it was already mentioned, it was recently detected in mainland Europe, and that's Greece, and basically that could mean the beginning of the new invasion. The problem is that the pest is highly polyphagous and it won't be limited by host availability, and can also fly long distances seasonally, which makes its management quite a challenge. It has also been a subject of regulatory attention in Europe, and it's classified as a priority pest due to its potential economic, but also environmental and social damage. Here you can see the occurrence records we compiled across the globe with its invaded range and its native range, which is the Americas. Here we used a climatic niche model, the so-called Climax model, just to obtain the climatic suitability across the globe, and of course in Europe in the end, which is our study area, based on the climatic and developmental requirements of the pest. And you can see that basically the yellow to red gradient is where the regions where the climatic conditions are suitable for permanent establishment, whereas the blue gradient highlights the regions where the pest can fly their seasonally established temporary populations and cause damage. And this damage can be as substantial as the damages in its permanent range. The issue of having identified suitable pockets in the south of Europe for the permanent establishment of its populations is that seasonally, this is what happens in its native range, the pest migrates northwards to either avoid summer temperatures or access more food. And this migration could happen in Europe in case the pest permanently establishes at the area of potential establishment where the conditions are suitable. So what we did is that we retained the occurrence records in its native range that fall outside the area of permanent establishment, and we basically measured the distance of each one of these occurrence records to the closest area of permanent establishment. And we obtained a distribution which we extrapolated into Europe. And basically, one could tell that it is quite likely the areas that fall within the 50th percentile of this distribution that could be invaded every year in case, the pest spread further in Europe. On the direct economic impact, we focus on grain maize, which is the main host of the pest. And we conducted an analysis based on the partial budgeting under a no-control scenario to, in a way, obtain an estimate, an upper bound estimate of the potential direct damage costs. And we see that in terms of gross margins per hectare, it seems that the countries that would bear the most risks is the southern European countries, namely Greece, Portugal, Italy, and Spain. But when we scale by the area of grain maize cultivated in its member state, the picture looks somewhat different, meaning that we can see that France, Italy, Spain, and Hungary are the ones with more, with wider areas of cultivated grain maize. And in terms of millions of euros per year, the average annual damage cost could reach up to 900 million euros per year in a very worst case scenario. Moving on to Phytophthora ramorum, this airborne pathogen that is pertinent and very relevant to both the forests, our forests in Europe and the nursery sector. You can see here the reports, the countries that report the pathogen, and the fact that it has been also for 20 years now a subject of regulatory attention as well. And we basically try to approach the issue in order to assess its potential damage costs in a similar manner. We use Climex, but in this time we focus on only the forests, and we retain only the occurrence records that were derived from forests and not from nurseries, because in nurseries the conditions could be controlled. So we just focus this study on forests, and we focus on two very important host trees, namely the larch and European beech, different larch species. And we can see here that the more or less 10% of Europe, of the study area, is climatically suitable for disease expression, meaning that the conditions are optimal and symptomatic infections and tree mortality are expected to occur. And of these 10%, the 42% is basically represented by the UK and Ireland. You can see here the areas are basically mostly concentrated in very wet conditions. This figure is just to give you a glimpse of the distribution of the two hosts we chose to focus on. This is the larch and this is the beech trees. And in terms of the potential damage costs, for 3 different mortality scenarios and 3 different spread rates, we can see that in both cases, the annual direct damage costs under a worst case scenario raise up to 100 million euros per year. Looking at the large trees, the UK, Austria, and Italy are the ones that bear the highest risks, whereas for beech, Italy, Germany, and Austria are the member states that are expected to undergo the potentially the highest damages. The last part of my talk is about the adoption potential of the VOC sensor among European nurseries, and this is basically the motivation is that at border controls, the phytosanitary inspections, as Andrea mentioned also, might fail to detect asymptomatic infections or infections that are latent. So actors that are involved in plant trade can be very helpful in reducing the risk of additional infestations that are coming into Europe. And nurseries can be one of these actors. So they are the ones that they have a direct interest in trading healthy plant material, but at the same time, they're not mandated to inspect on their premises the important consignments. So we basically we surveyed 343 nurseries that were distributed in Italy, Romania, Germany, and France. And here you can see the sort of what type of plants they are mostly cultivating, which are ornamentals and fruit and vegetables for the most part. Roughly a 56%, looking on the average pooled sample, reports that they always perform phytosanitary inspections in their premises, but at the same time, the rest roughly say that they actually rely on the phytosanitary certificate that the consignments are issued with. The methods that they follow to perform these phytosanitary inspections are mostly visual, which can be quite fast, but could also be inaccurate. The visual inspection is also what mostly is done at border control level. And some nurseries reported that they could send samples to an outsourced and external laboratory for further inspection. The participants, the respondents of the questionnaire, were invited to make selection, and this is an example of a choice card. So basically, we listed different characteristics of the VOC sensors, namely the time to receive results, the detection reliability, how much it costs to inspect one batch or a consignment, whether they are able to certify the fact that they further inspected the consignment in their premises, and how they prefer to access the VOC sensor, meaning that whether they prefer to buy it for their own nursery or share it with another nursery, or just outsource it as an external service. And the participants, they were invited to make a selection every time. There were 8 choice cards presented to them between option 1, option 2, that was representing different VOC sensor configurations, and the current practice, basically to not adopt, that they are not willing to adopt the sensor. And this is a glimpse of the results. So basically what this table suggests is that the cost per inspection is always negatively and significantly affecting decision adoption decisions towards the sensor. So every country cares about the cost, which is an expected outcome. And when we look at the time to receive results, it's also a very important factor, except for Germany, that Germany seems to not. Basically, the range of this attribute was 1, 2, and 4 hours. So one could say that Germany maybe doesn't really get affected by the time to receive results, because it's still the same day. Usually when they outsource the service to perform further diagnostics, it can take more days. So moving to detection reliability, everyone cares, and the more reliable the sensor, the more the predicted probability of adoption, except for Romania. But Romania cares to certify that they perform further inspections in their premises, as well as France. Again, Germany, the certification potential plays a significant role to Germany on adoption decisions, but it's negative. And this could mean that they perceive the certification potential as an additional paperwork that will hinder their workflows in a way. We see that the ownership structure doesn't really play a role. And in this point, I want to say that for more information, you can access the PurPest Work Package 5 deliverables, which we all list, which includes basically the assessment of all 5 target pests and more information on the survey we did. And also, we have 2 publications that are pertinent to the first deliverable, 5.2. And you can find these on the PurPest website. Thanks a lot for the attention.
Thank you very much, Stelios, for reporting about the economic damage potential these pests cause. And as you nicely have pointed out, the economic damage potential is extremely high. So there is a lot of benefits to be gained from having a better control of the invasive species that are investigated within the PurPest project. Now I like to open the floor for questions and comments from the audience. And looking at the Q&A, there was one question that has been raised by Simonetti, and that is addressed at Thomas. She would like to know more about what are the ornamental plants that are affected by Phytophthora ramorum. You have mentioned rhododendron, but there might be also other plants. Maybe you can elaborate a little bit more, and perhaps say also something what we can do as individual plant buyers if we can do something, or maybe not. What your take is on that, Thomas?
So the main hosts in the ornamental trade worldwide are rhododendrons, without any doubt, but also viburnum plants, Pieris japonica, camellias are highly susceptible and highly contaminated, but it's a wide range of plants that can have Phytophthora ramorum. And the problem with Phytophthora is that also non-host plants can actually be infected in the soil, at least, with Phytophthora ramorum, and any of the other 124 invasive Phytophthora species we have up to now provenly, evidently introduced into Europe. They can be always in the soils of nursery plants without doing any damage, because they are very often anyway suppressed with fungicides. And that's why they don't show disease expression, but they are present in the soils. What can we do at the moment? I don't recommend to anyone to buy any plants from any nurseries or garden centers, because the infestation rate with Phytophthora across Europe is almost 100%. 100%. With one of 124 or maybe 5, 6 per plant species even that you get. And the bigger the plants are, the more likely they were already in several nurseries in the grounds. So a big ornamental tree that you plant in your garden bed could have been in Italy in a nursery, and in Netherlands in a nursery, and in a German nursery where you in the end bought it or so. So it is, at the moment, it is a huge risk. And as long as we don't have a nursery accreditation system with proper controls and proper growing conditions to produce plants in a biosecure way, I cannot recommend to buy any plant from anyone in Europe. And I'm always asked, don't you know any nursery that is more reliable, trustful or so? And I say, no, you can't, because they all buy from the big producers. So they're all infested in the same way. And some of the very small nurseries, they actually don't produce anything themselves anymore. So they order via the internet from 20 different big growers Europe-wide, and they can have 40, 50 Phytophthora species in their small nursery. It's not uncommon. So I cannot recommend any nursery buying at the moment. It's very sad.
Oh, this is very sad news also for the nurseries, right? If you recommend, don't go to the nursery and buy.
No, that's actually for the nurseries at the moment. This is a huge business because no one knows it actually. That means that people buy these billions of plants every year, and half of them die in the next 2 or 3 years in the gardens, and then they buy new plants. So the nursery doesn't have to wait 10, 20 years to sell a new plant. They sell it already within 3 years. So as long as the consumers are absolutely uninformed, and they are kept uninformed deliberately by the governments, and also by the phytosanitary services, which in my opinion is almost criminal. As long as they are uninformed, this is the best business nurseries can have, to sell plants that die within a short time. Sadly.
That I would perhaps also agree with that. This is news that it might not be so welcome to the nursery owners as well, in the sense that, right, if we provide more information, then their business model might be also substantially affected. So there is an interesting trade-off within this kind of issue related to invasive species and Phytophthora in particular. I think that really needs further discussion. So thanks a lot, Thomas. And now let's have a look at the other question, and that is by Joao Godinho, addressed at Jana Kollatz. For pests like brown marmorated stink bug, are the biological control options that you have mentioned in your presentation already available in the EU market? Jana, can you say a few words on that?
Yes, I can. Unlike conventional plant protection products, biological control agents are not regulated EU-wide. So if you ask for the EU market, that's a bit difficult to say. You would have to look into your specific country. But what I can say is that there is a native species, parasitoid, which is called Anastatus bifasciatus. And that species is on a list from the Mediterranean and European Plant Protection Organization, listed as a species that is used in at least some European countries. I know of Italy where it is marketed and sold, but it might be also available in other countries. And it is a native species. And once it's on that list, it is relatively easy also for companies to register it usually. And then there is also an exotic species that first has been introduced unintentionally into Europe. The species is called Trisulcus japonicus. It has probably been introduced accidentally the same way like the brown marmorated stink bug itself. But it is now present in several European countries such as Germany, Switzerland, and Italy. And at least in Italy, I know that it has been also released intentionally to be used as a biological control agent. And it may have been released in other European countries as well, but this information is a little bit difficult to obtain.
Thank you, Jana. There's been another question has been raised by Irene Donati. For polyphagous insect like the brown marmorated stink bug, do you expect that the VOC profile to be plant dependent? Maybe Damien, you might be in the better position to answer that question.
I'm not sure if I'm the one, but
okay.
Yeah, no. But it seems that yes, the profile there seems to be quite different from one plant to another, and also sometimes, for example, for the maize plant, we realized that one molecule was very interesting because it could be used to actually differentiate the healthy and infested plants, while the very same molecule could also be emitted by other plants, whether they are infested or not. So then, of course, what can be also very difficult is that at the moment we study a lot, let's say, single plants, but it's true that in real environment, then we know that the mixture of all the VOC profile emitted by all the different plants will make things a little bit more difficult. But yes, the VOC profiles are very different depending on the plants.
And do you expect that you will be able to differentiate so that you basically can say, okay, this is a brown marmorated stink bug, and that can be detected on plant X, Y, Z?
Yeah, but this is why all this machine learning is also implemented into the machine, because we need to, or the user will need help to be able to differentiate this. The difference between the different plants, and also again what we see for most of the plants is that we don't have individual tracers, so we will have to look at all of them at the same time, like the whole profile, and we have good hope that this whole profile will help us to differentiate between infested and non-infested, even in very complex environments.
Okay, thank you. There's another follow-up question, and that is, through the use of these VOC sensors, what are the insights related to potential reduction of overall pesticide use? So basically, can we expect that if we have these sensors and they work, for example, at border control or at nursery, etc., that this then in the end may result in reduced pest control methods being needed? Maybe Andrea, you can respond to that question.
Yeah, I think you can also ask the other way around. How much pesticides are you going to use when you have Europe invaded by all these pests completely? I think if you think about that, what is the cost of not doing something? And I know that's often overlooked, and they think, oh, everything's fine. Yeah, all is fine until it's too late. And so that's, I can't get, I am 100% sure that by reducing the risk of having these plants invading Europe, we will save pesticides. You can now argue that maybe in a couple of years, we will not be allowed to use any pesticides at all, and then, what's the difference? But then you will have a huge loss due to these pests. And the other thing is that you could also think about using the sensor for site-specific pest management. So if you have something out there on your maize plants, like Amandine showed, you could spray not the whole field, but maybe just those parts of the field that are actually infested. Now, if plants, if insects fly away, that's a bit more difficult. But if you have pathogens or if you have nematodes, you might be more specific towards that. So instead of using 100% for the whole field, you might only need to have 20%. And that's been shown for site-specific weed management, that you can significantly reduce the pesticide input.
I also look at Stelios catarrhis. So Stelios, we did look into the no control scenario and what this may mean. So what do your results tell us about the potential for reducing pesticide use, or the pesticide use increase that we can expect if we do not control the species?
Yes, thank you, Justus and Andrea. Just to add something on this, basically, in a way, the site-specific management can be also, used to prevent or manage the pest on a regional scale or on the specific location. But it's also important to know that your farm, let's say, is infested at an earlier stage, because the more the time passes, the harder it is to control them and more expensive it is to control, of course. So the potential that the VOC sensor gives to farmers, to end users, to know that something is infested before the symptoms occur, before it's too late, so to say, is also could be a big economic benefit when looking at the time of management, because it also, after, that's the whole narrative also that Europe proposes, that prevention is always the cheapest strategy. So the sooner we can act on the management scale, the better and the cheapest.
Okay, thank you, Stelios. Andre, you also have raised your hand, so you like to comment on this as well?
Yes, just a small comment, because in the case of the fall armyworm, for example, the European Union recommendation, or how do you say? If you have a fall armyworm detected, you have to spray your field and your whole field, and even dozens of kilometers around your farm. So even if we come up with this sensor technology that enable us to just spray very targeted amount of pesticides on the plants that are infected, the regulation have to change in order for us to put that in place. Otherwise, this will not happen. So it's just to say that it's not only this research question, it also has to be taken into account at the European agency level for the regulations to change in this way.
Yes, Andre, many thanks for this very important remark here, that this needs to be taken into consideration. We are not operating in an apolitical world, and that this may also be something that needs to be addressed if we want to be successful. I see that Thomas has also raised his hand. Is this directly to this question, Thomas?
Yes.
Then please be brief, because there are a number of other questions, and I think we should address them as well. Thomas.
A quick comment: there are no fungicides available in Europe to eradicate Phytophthora from soil. It's impossible. And there's also no fungicides registered anymore for fighting Phytophthora ramorum in natural environments like forests. That means that makes it even more pressing to prevent any further introductions of other Phytophthora species to Europe with good sensors.
I think, again, a very important contribution here, that even if we would allow to be using pesticides, plant protection products, there are currently no plant protection products available. And that further increases specifically for Phytophthora ramorum the need for having better control methods available. This is then also related to the following question that we have from Lynn Lawrenson. Can the sensor look for multiple pests, or are the programs running specific to each pest? And how easy is it to expand the system to include more pests. So I relate this again back to Damien. Maybe you can enlighten us on that.
That's for me. Now in the project, what we try to do is to have rather a single method, even though it takes slightly longer for the analysis. So a single measurement will take 30 minutes, but we try to look at the broader spectra as possible, so that actually as let's say the single machine or the same machine could be used and be shipped at different places and still be able to actually identify the different pests. And then maybe in the next step of the project, when the machine or the system will be evaluated for each pest, we might succeed to reduce the analytical time and make it more specific for a single pest. But at the moment, we are more like targeting everything with what we call the cycle time, so the time needed for one analysis of like around 30 minutes, and which is quite a lot, but we hope that it will allow us perhaps to see most of the pests with a machine which would be quite similar for all the evaluations. And so we hope, of course, that this configuration will be possible or could be adjusted for other pests in the future.
Right, talking about the future, I'd like to forward this question to Andrea about what may be the next steps that are going to come for making the strategy more successful. So maybe Andrea, you can enlighten us a little bit about what is going on there.
So in PurPest, we have a proof of concept, proof of principle. Does this work or does it not work? And that's what that's basically what we're focusing on. And we're not going to have a ready to go tool in the end, but we have a follow-up project that's called SensorPest, and that's also a Horizon Europe project where we are focusing only on the Phytophthora and Phytophthora and on the brown marmorated stink bug and the Japanese beetle. And there we are first also looking at single infections, but also then on co-inoculations, so that we will have two different Phytophthora on one plant and see if we, how is the profile changing? Can we detect them as well? Are there similarities? Are there not similarities? So the future is really to generate a tool that can be adaptable to several different Phytophthora species still within the same genus, but we have so many Phytophthora that are damaging that we thought it would be really important to have a tool that can self-learn with the help of artificial intelligence to detect those pests, and especially the Phytophthora that we have already identified, but that also can learn based on the information that we give them if there's a new Phytophthora coming into Europe. So the future is basically going in that direction, that we are trying to be highly specific, but also broad enough that we can anticipate the future threats that are coming to Europe.
Yes, Andreas, thanks a lot for pointing this out. So there is a follow-up project that tries to further develop the technology being available. And we have seen here in this presentation and in the discussion how important this is to become more specific, and how important it is for biodiversity, for appropriate pest control, having technologies available that allow us to specifically identify the issues that we are facing. The issue with Phytophthora ramorum just illustrates us that how difficult it is and how important it is to get a control on this invasive species, looking at the potential damages and the damages already been done. So please stay tuned on the further developments within the PurPest project and the so-called follow-up sister project, SensorPest. And there was the question by Ariba Lopez, thanking us for the excellent presentation. I fully agree on that. We will make these presentations available via the PurPest website. So please have a look at this, and we will inform all the participants about this. But before we close this webinar, I like to hand over again to the project coordinator, Andrea Ficke, for closing the session, so to say, with some hopefully encouraging words for the PurPest and the VOC technologies.
Well, thank you very much, Justus. Well, I'm very glad that there are many people that were interested in this, in the challenges that we're facing with importing plants and unfortunately also pests and pathogens. I hope that everybody of you, who's now here and that has maybe not just start thinking about that, you can always contact us. You can give us your input, because we really need to know how can we tackle this together. I mean, scientists can only do so much. In the end, it's actually going to be the users and the people out there at the nurseries, at the import stations, at the authorities. They're the ones who actually have to make this work. We can only deliver expertise and tools, but put it in action and make the difference. That's all of you. So I think with that in mind, I thank you again for joining us today. Keep in contact, talk about the concept, tell other people about the challenges we're facing with plant imports. And if you feel that there's something you want to contribute, that some thoughts that you have and that could relate to our project, contact us. We're very open for that. Thank you very much.
Thank you too.
Yes.
And with this, we finish the webinar. Thanks a lot to all the speakers for a fantastic contribution, and to the audience for staying with us, and hopefully to see you at the next webinar of the PurPest project. Thanks a lot, and bye-bye.
Thank you.
Thank you. Bye.